We left off last time talking about alternative splicing. And you got the basics of what you needed to know, at least for splicing. But I wanted to talk about splicing a little bit more and another thing called RNA editing. The beauty of eukaryotic genomes, like your cells are, is you've got these introns put in them. And at first they seem superfluous. Why would you have introns, this inner mixing region that's not for what you actually want to code protein for? This comes back to this idea, and I asked you on the exam, what the central dogma of molecular biology was. When Watson and Crick discovered DNA, and we started realizing that DNA coded for mRNAs and mRNAs coded for proteins, the dogma was, for every protein, there's a place on the genome, and that's the only place proteins come from. For every one place on the genome, there's one protein. And if you want a different protein, you have a different location on the genome for a different protein. Well, when we discovered splicing, we realized that that's not the complete story, right? There's not just one single location on the genome might actually code for multiple varieties of proteins because you can alternatively splice them, right? So I could have a genomic region that gives me this nuclear RNA, this pre-RNA that still has its introns in it. But that doesn't mean I just splice out the introns and I get just one protein product, right? I can alternatively splice, so one location on the genome could give me two protein products in this simple case, right? If I've got two introns, I could splice both of those out and get all three exons, exon 1, 2, and 3 put together. That would get transported out into the cytoplasm and I make a protein off of it. Or I could delete the second, right? The introns give me the option to edit the RNA. The cell can decide what type of protein it wants to make off of this mRNA. And there's more elaborate things here, right? So you could do, this is called the cassette type, where you could either insert it or eject it, right? You're either in or out, right? You can also do alternative sites. So we'll do mutually exclusive is the next example. This is where the option is not to just put everything together. It's an either or question, right? Either you can put in, either you put in 8 and you splice out 9. So it's mutually exclusive. You get 8, or you can splice out 8 and include 9, either or, right? There's actually really important biology that's determined by these changes. In your Drosophila, the difference between a male and a female Drosophila is alternatively splicing of one gene. It's called double sex is the name of the gene because it codes for both sexes. So if you splice double sex one way, you turn on the patterning for developing as a male. If you splice it as the female way, then you become a female. So I mean, there's other genetic factors. They've got X and Y chromosomes, but it kind of narrows down to that, right? How do you splice double sex? That's what tells you to become male or female. There's also really important functional differences between cell types. One cell type will splice a protein, or splice its mRNA to make a certain protein. Another cell type will do it in a different way. So depending on if you're in a liver cell or a kidney cell, you might express the same protein, but slightly different variants, okay? Which gives you wonderful plasticity, right? We can do all kinds of interesting things. There's even more elaborate splicings where instead of just splicing the entire, so in this case, I've got 3 exons, instead of just splicing out the entire thing, you can either take the whole thing or you can alternatively splice at the 5' end and just take a little bit of the exon. This can actually have really significant changes if you start messing around with how many nucleotides got included, which we'll talk about in a minute. But this could actually, this is just kind of in or out, right? This is this one, 8 or 9, one or the other. If you start alternatively splicing in the middle of an exon, that means you could start changing the frame. You could actually read the entire sequence in a different frame, which we'll get to when we get to translation. But this can have really significant, really fundamentally different proteins that get made off of this. So you can alternatively splice on the 5' end. You could alternatively splice on the 3' end. So basically you can either take the whole thing or a little bit of the 5', or you can take the whole thing or a little bit of the 3', right? It's basically the same mechanism, but it depends on which piece you get to take. So yeah.
Are introns always spliced out?
Yeah, introns are always spliced out. I guess I should say that as a general rule. We call it an intron because it's a thing that gets spliced out, right? Now, maybe there's things that we're currently calling introns that we shouldn't, because maybe sometimes they stick around, sometimes they don't. So I guess that's kind of just a semantic issue. But usually when we say intron, we mean a thing that doesn't contribute to the protein sequence. Okay? So we might be wrong about that, but that's just the definition of intron. Okay, so that's splicing. It's going to give you a host of variety. And think about a protein, think about a gene that has 15 introns. And at every single one of those 15 introns, you could alternatively splice. That gives you exponential numbers of proteins that you could make off of that, right? Because if it, intron 1 and 2, you splice this way, intron 3 and 4, you splice this way. If you've got 15 of them, right, and every single one of them has all 4 options, that's a lot of different proteins you can make. Okay? Some human proteins don't have, or some human genes don't have introns. It's pretty rare. Almost all of them do. Some of them have, like I said, 33 exons or more. I don't actually know what the average is. That would be interesting to look up, but on average, you've got several. So, yeah, Rachel.
Does that have anything to do with genome size as well, like the more introns, the smaller?
Yeah. So it was actually, so the question is, does that have anything to do with genome size? Yeah, it has a really big thing to do with genome size, and actually gene number. When we started sequencing genomes, we didn't know how many genes organisms had, right? We'd done a lot of biochemistry, we counted out the proteins, and you thought, okay, well, based on the number of proteins that we see, we estimate we're going to have this many locations as distinct places on the genome. And it turns out we have much fewer gene locations on the genome than was predicted by how many proteins you have, right? Because a lot of these overlap. So when we started doing genome sequences, you're like, well, you've got a fly and a human, and they have roughly the same number of gene locations on the chromosome. But a fly and a human are vastly different in terms of their organizational complexity. So it didn't match on well. Every trait you have is not coded for by one protein that exists on one piece of your chromosome. Right? And then there's also widely varying things too. There's organisms that have ridiculously huge genomes, but then there's a very similar morphological, so you could take like one fish and another fish, and they're both fish, they do basically the same things on kind of an organismal level, they're kind of the same complexity. But one of them has 4 times as much DNA in this genome than the other one does. So yeah, complexity and genome size, we thought were going to map onto each other, and they don't. So yeah. Gene size. We haven't gotten quite to gene complexity, because big genomes can be very complex, but they're not complex for the reason of one gene coding for one protein. Right? It's more intricate than that. So, Will and then Josh.
How does that relate to when scientists will say that your DNA is similar to that of another organism, and they give you a percentage or something like that?
Yeah.
Are they saying that that all of your DNA is this percentage similar, or the coding part of your DNA is?
Yeah, that's a really good question, and I don't know the answer because a lot of different cases.
Okay.
So, you could say on a nucleotide level, how many nucleotides actually match up, and that would be one way to say how similar they are.
Okay.
But you could have all kinds of nucleotides similar, but things rearranged all over the place on the chromosome. Or you could have nucleotides similar, but one has 4 copies. There's actually, I'm not sure of good ways to really make those percentage comparison numbers, because, are we comparing the entire thing? How many repeats there are, how long the chromosome is, or just the coding regions? Typically, it's been just the protein coding regions. We compare those because that was thought to be the most fundamental thing, right? The proteins is what determines your phenotype, so we kind of just don't care about anything else in the genome. We're just going to compare protein sequences, coding sequences. It turns out much more of the genome gets made into RNA than we thought, and that RNA is doing things. So we should start thinking about all those, what used to be called junk DNA. Everything in the genome that didn't code for a protein was kind of thought of as being junk, right? It just doesn't code for anything, it's just repetitive, it's redundant, it's not doing anything functional. It turns out that like 80 to 90% of the genome is actually made into something. And a tiny fraction of that is actually made into protein. Most of it's made into RNAs. So we're going to talk about RNA in gene regulation, about what it's doing. But genomes ended up turning out to be much more complex than just DNA making an RNA, mRNA, and that making a protein. A whole lot more going on. We've seen some of that already in terms of the promoter regions, right? It's important to get RNA polymerase sat down, and that kind of coordinates when that gene gets made and what tissue it gets made, but there's much more to uncover, so we'll get to it. So we'll do Josh, and then we'll move on.
Well, I think it's kind of a similar question. Like, I was just asking more like, how does it kind of determine, like, if it's determining like this is an intron, this is an exon, and kind of like splicing that out
Yeah.
to make the protein. Like after that, and like what happens to all the introns?
The introns just get degraded, and the nucleotides get reused.
Okay. How does
Now,
it, like, determine which?
in general, that's what happens. Sometimes introns are actually really important pieces of RNA that go back and regulate when genes get turned off and turned on.
Okay.
So yeah, they're feedback mechanisms as well too. So they're, even though you think of, well, okay, those introns, we just showed them in black, they just get spliced out. They don't all just disappear, right? That might actually have some role to go feedback and say that's a gene that got made and maybe help turn on another gene, or feedback on itself and determine when that gene's time is done. So yeah, there's lots of stuff going on with the introns.
Okay.
So that's not junk either.
Oh, gotcha.
Yeah.
So it bases like the exon based on need, like I need this kind of.
Yeah, I guess so, based on the cell's need.
Okay.
Yeah, or who the cell is. Yeah. Yeah. Okay. This will probably just bring up more questions, so this is good. On another level, RNAs can be edited, mRNAs can be edited. So you can alternatively splice them, but there are mechanisms also that actually change the nucleotide sequence of the mRNA. The way you determine the mRNA sequence in the traditional molecular biology dogma is whatever's written in the DNA, you make an exact copy of it in an mRNA, and then you can alternatively splice it, but that's the dogma, right? The DNA just told the mRNA what it needed to be. Well, it turns out we actually edit mRNAs. Your cells actually edit them, and will change the nucleotides in the mRNA to code for different things. There are two mechanisms, two general mechanisms of this. It's either template-derived mechanisms or non-template derived mechanisms. And the example here in this illustration is a template-derived. Okay, so here on the bottom is an mRNA. Okay, that's a new mRNA that just got made off of the genome. RNA polymerase made it. It gets exported out into the cytoplasm. But there is another complementary piece of RNA that's also been made off of the genome. And when it's involved in editing, we call it a guide RNA, gRNA. So it's got complementary sequence. It might have been made off an intron. It might have been made off the reverse complement. It might have been made off of some other completely distinct region on the genome. But it's a short little piece that has complementary base pairing ability to your mRNA. So this will recognize it and stick down. But right here, this bump is where the guide RNA and the template mRNA don't have the same nucleotides. They're differing in the nucleotide. So they don't make traditional base pairing, they don't stick down perfectly. So you get this little, very, very small little piece of denatured single-stranded region. And what happens is this editing complex called the editosome comes in. This is a bunch of proteins, and these could be various different proteins. So it's not the same group of proteins in every single cell. It varies from cell to cell. Comes in and will actually determine that the mRNA needs to have its nucleotide changed. So the guide RNA tells the editosome that nucleotide needs to be changed. Right? So that one will be cut. There's like endonuclease activity, breaks the phosphodiester bonds, recruits in the nucleotide that will base pair up to the guide, reforms the phosphodiester bonds, and now you've actually changed the protein sequence that's going to be made in the cell. This is completely undetectable if you just take genomic DNA and sequence it, right? The genomic DNA told you that that's what the sequence was on this mRNA, and that's what's going to code for the protein. But this editosome can come in, change the nucleotide, and now we've changed the protein sequence. Sometimes this is subtle shifts in just one amino acid change. Sometimes this is inducing a completely new stop codon, and you make an entirely smaller, truncated version of this protein. So it could actually have fundamental changes. Sometimes it actually removes a nucleotide or inserts an extra nucleotide. And that can then shift the entire frame that you read the protein in. And instead of reading it in 3 basis pairs and that coded for a certain protein, well, if I stuck another nucleotide in there, I've shifted the frame now. And every single amino acid after that has been changed. So we'll talk about frame shifts again more, and that should be clear. But you should know it can either swap it, change out the nucleotide, or it can completely replace it. So you can have single amino acid changes, alternative stops, or if you induce a frame shift, after the frame shift, you're going to change the amino acid sequence of every single amino acid after that one. Here's an example of an alternatively edited gene in your body. There's a gene called apolipase B. It's a protein that holds fats. It's a lipase, lipoprotein, protein that helps transport fats around in your tissues. It's in the liver. It's not edited, so it creates a 100 base pair, 100 amino acid long protein in the liver. In the intestines, that same protein gets expressed at the mRNA level, but it's edited. There's a premature stop codon that tells it to stop making protein, or stop polymerizing amino acids earlier. And so you make a half the size protein that does a slightly different function in the intestine. This is pretty amazing. We've only learned about this in the last couple of decades, that this even exists. And it changes the whole way we start thinking about how cells run, right? The cell is determining what type of protein to make. The genome is not, right? I mean, in a way, the genome, in the template model, right, that template tells you how to change it. But you had to turn on that template, get that made into RNA, and then also what proteins you put in the editosome change that. So it's kind of, this is called epigenetics. This is like, instead of the genome determining what the protein is, it's the cell determining what the genome should make in terms of protein. This is like extra DNA programming, modifying the genome. So.
It seemed confusing too just now. You said that you would think that it's the genome telling the cells what to, but it's the other way around? Or sorry, maybe I'm just missing.
Well, I mean, the cells in the liver or cells in the intestine are determining what protein gets made, right? And the genome in the liver is the same as the genome in the intestine, right? They've got the same exact genome, but the cell type is determining what protein is expressed, right? That's what I mean by the cell is determining it.
And does that happen also with, I'm trying to think of an example where only on certain occasions you would make a particular protein. Is that like what you do with people like lactose? Because lactose is constantly made in your system. Is that the same idea?
Well, I mean, it's an additional way that cells differentiate from each other, right? So you can either turn on a gene or not turn on a gene, right? Or turn on a gene for a short period of time or extend the long period of time, right? So that's the expression.
Okay, so it's different.
This is different. Both of these are being expressed, right? They're expressing mRNA in them. Both the liver and the intestine are. So this is like a downstream version of that. Once you've decided that this is an appropriate gene to turn on, now what version of the gene do you want to turn on, right? So it's editing which kind it wants. So, yeah. No, it's okay. This is fun.
But, so I guess in terms of like glucose lactose intolerance, could theoretically we come up with something that would go in, like, do those people have mutations in actually their genome typically, or is that protein's not being turned on?
Lactose intolerance, everyone tolerates, well, almost everyone tolerates lactose as a baby, right? You're drinking milk almost entirely. And we're actually in the process of weaning our baby off of milk. So you only become lactose intolerant when you stop making the enzyme that can digest the lactose, right? And actually, if you survey the entire world population, most people turn that gene off, beginning to approach adolescence. is where it kind of tapers off. And that's where most people start to think they've become, well, if you're going to become lactose intolerant, that's usually when it becomes, right around maturity, right? And that's your body's way of saying, we don't need milk as our primary food source anymore. You can start eating food, right? The weird people are actually us Caucasians who, for whatever reason, continue to express that gene late in life. So, you know, I can go have a huge glass of milk followed by some cheesecake that Tamara just brought and digest all that lactose just fine. But that's an anomaly actually in most people in the population. Most people in the world actually stop making lactose. You can't digest the lactose, so the bacteria in your gut happily chew on that lactose that you can't metabolize, and then you get all the gas and the side effects of it. So, yeah. Yeah. So, if you're lactose intolerant, hopefully that makes you feel better. Like you're in good company with most of the rest of the world. So, all right. So that's editing. We'll talk more about frame shifts and mutations and how that affects proteins after we get through making proteins. So I'm going to shift and we're going to talk about translation now. The mechanism of how nucleotide language gets translated into amino acid language was by and large figured out by these two guys. One you already know, Francis Crick. The other you may not know. This is Sidney Brenner. He was a South African born. I think he did most of his work in the US, but these two guys worked out actually in a little bacteriophage, a little virus that infects bacteria, what the actual nucleotide sequence is that gets translated into amino acid sequence. So they worked out what we call the genetic code. This is the genetic code. Well, one of the genetic codes. There's actually multiple genetic codes. NCBI, the National Center for Biotechnology Information, has a list of all the versions of the genetic code. So some bacteria, archaea, mitochondria, a bunch of different organisms actually have slightly different genetic codes. So the genetic code, sometimes you hear an argument for common ancestry is, well, every organism on Earth shares the same genetic code, so they must be all descendant from the same organism. That's not actually true. There's actually multiple, I think there's more than a dozen different versions of the genetic code of what the nucleotides actually code for in terms of amino acid. This is the mammalian typical traditional genetic code, okay? So we're translating, turning one type of language into the other. People had postulated that it must be 3 nucleotides that make up the genetic code because we already knew, well, there's 20 common amino acids, and if you only have 4 bases in 2 positions, that only gives you 16 different combinations. So that can't account for all of the 20. So people said, well, it's probably 3. Crick and Brenner are the ones that kind of worked out what 3 code for what amino acid. They had help with other people, but they were the drivers behind this. So here's the nucleic acid. It's written in RNA language, so there's uracils instead of thymines. But the genetic code, in DNA language, those would just be T's. So UUU codes for phenylalanine. Now, UUC also codes for phenylalanine. The genetic code is what we call redundant, or sometimes called degenerate. And it doesn't mean it's bad. Degenerate is not a derogatory term here. It's just saying it's not containing as much information as it possibly could, right? UUU and UUC could code for different amino acids. They're redundant. They're degenerate, right? We've just collapsed those into one thing, right? They both code for phenylalanine. Notice that it's the 3rd base that often is the difference between them, okay? The first 2 bases will almost always be the same, UU and UU for phenylalanine. It's just the 3rd base that might change. And we call that the wobble factor. So it can wobble in the 3rd base. Here's leucine. Leucine, you could wobble any way in the 3rd codon, or the 3rd nucleotide of the codon. And so we call the 3 nucleotides the codon. And I think was there another leucine around? There's 4 here, right? Am I missing one?
Is that not leucine right
Oh, yeah, I'm sorry, right above it. Sure, there it is. Okay, so leucine, CUU, the U can change to any one of the nucleotides. Still codes for leucine. If you have a first letter change, it's usually an entirely different mechanism, right? So leucine is actually very common. You can make it a bunch of different ways. Notice it's going to wobble here in the 3rd position, or I'll have a completely different code, and then that wobbles just in the 3rd position, right? So.
Is it the second codon then that kind of holds the code
No,
for leucine?
I'll show you why this is true in a minute, and it should answer that question. Okay. It's having to do with the tRNAs that bring in the amino acid. So, yeah.
I don't really want to know the answer to this, but are we going to have to memorize this?
No, you do not have to memorize this. Yeah. No. I don't have this memorized, nor do I think it's important for you. If you need to do this work, there are computer programs that do it, or you could just print this out and do it yourself, right? So yeah, don't memorize this. The one thing you should know, though, is the ones I've highlighted here in colors. Those are the only 3 that you need to know, because these are the really important ones that tell you something about a gene if you're reading DNA sequence, okay? The methionine, there's only one way to code for methionine, and it's AUG, at least in this genetic code. There's alternative ones if you're talking about some bacteria or other organisms. But in eukaryotic, multicellular animals, AUG is the only way you code for methionine, and it is always the first amino acid that is made. So it's always the start codon. So if you're going to turn an mRNA into protein, the first codon is always the AUG. Sometimes it gets edited, and it might not always be the first amino acid that ends up being in the protein, but typically when you actually start making the protein at first, the methionine always the start. Okay. Now, probably if you search the literature, you can find exceptions to the rule, but almost always it's the methionine. The other 3 are the stop codons, and I want you to know what those ones are. UAA, UAG, and UGA. That's going to tell you where the protein stops adding amino acids to the end of it. So you should know the starts and the 3 possible stops. Okay. You can already kind of start thinking about mutations, right? If I have a UGC, I'm only 1 mutation away from a premature stop codon, right? Same with if you're coding for tyrosines, right? You're only 1 amino acid away, or I'm sorry, 1 nucleotide away. Okay. So I have 64 different codons possible. I only code for 20 amino acids and 3 stops. The other ones are redundant. All right, how is the genetic code read? It's read because transfer RNAs read it. Typically we say what makes proteins? The ribosome makes proteins. Well, the ribosome is kind of like DNA polymerase, RNA polymerase. It just does the chemistry of putting them together. What actually determines what amino acid is going to go there is the tRNA, the transfer RNA. Okay? So here's a transfer RNA. It's a single-stranded piece of RNA, 5' all the way to 3', and it makes this characteristic cruciform structure. It kind of looks like an X or a cross. The 5' and the 3', as it folds back on itself, is going to form these stem loop structures. And so you have loop 1, loop 2, here's a tiny little loop 3, and here's loop 4, and then it backs back onto itself. When you actually fold that in solution, it doesn't really look like a cross anymore. It kind of folds into this L shape. Okay. You should know that all of the tRNAs all have 4 loops like this. They vary slightly, but most tRNAs have very similar sequence in the part that's making the stems and the loops. Okay. That's not identical, but all the tRNAs, and there's dozens of tRNAs that get made. Okay. The part that is unique to each tRNA is the anticodon. And the anticodon is 3 nucleotides which should already give it away. This is the way you translate. You match up 3 nucleotides that are in your mRNA with the 3 nucleotides of the tRNA, and that's what tells you what amino acid to put there. Okay? This tRNA, this example, is got an amino acid alanine on it, and it's always covalently bonded to the last 3' nucleotide. Okay?
That's true for all of them.
Yeah, all the tRNAs will have their appropriate amino acids stuck to their 3' end, covalently bonded. Okay. There are 49, at last count, I think this is the human, 49 different tRNAs that have different anticodons on them. That's more than you would guess. That's because you have mitochondrial specific ones. The mitochondria has a slightly different genetic code than a human does. So there's 22 mitochondrial, and then there's, the difference of that would be the humans. I'm saying this for all posterity on the internet, and now I'm second guessing my numbers, but there's a lot of different tRNAs. They're all hooked up to specific ones, so take those with a grain of salt. I should have double checked that before I told you that specific. What I want you to see, though, is you can reuse tRNAs, and this is where the wobble comes in. Okay? So here is a leucine amino acid stuck to the 3' end of the tRNA. Here's the mRNA that's being read, and here's the codon. CUC codes for leucine. It's because leucine's always stuck on the tRNA that has the complement, the reverse complement on it. GAG, right? Normal base pairing, those hook up, and that tells, that's what makes the CUC code for a leucine, right? Now, you can also put leucines on, you've got the leucine on the identical tRNA, GAG, but CUU also codes for leucine on the mRNA. And that's because the base pairing at that 3rd nucleotide is not the most important thing of getting it down, okay? So if it can make the first 2 base pairs, then the tRNA is probably going to work. Same tRNA is probably going to be able to work on both codons, because that 3rd one doesn't matter so much. Okay. So the genome, the genetic code is basically two base pairs specific when it's not so important, and only when it's really important to have a different 3rd codon is it going to use a different tRNA. Right? So the genetic code is 2 nucleotides when it needs to be 3 nucleotides if it needs more specificity. Right? Okay. Now, if it's going to have, remember I showed you leucine had, I forget, UUA, I forget what it was. Go back here. To the genetic code here. Here's leucine. UUA, UUG, those could use the same tRNA because it's only the 3rd position that's wobbling. If I'm going to use these codons, CUU, I'm going to have to put leucine on a different tRNA if I'm going to use those codons. Does that make sense? Because the first 2 are the really important ones, so I'm going to have a tRNA that's specific to those first 2. Now that same tRNA could probably be used for all of those guys. Organisms have preferences for which codons they use. So you can actually, if you're reading an amino acid, if you're reading a gene sequence, you can sometimes tell what organism it is by which codons they're using for which amino acids. So for example, some organisms might use this group of codons for their leucines and very rarely use those codons for leucine. Other organisms might use that more common and not use these ones. So there's preferences that Even though it's the same genetic code, sometimes it's like dialects, I guess. You know, that word is capable of being used in both organisms, but in one dialect, we always use this word for that. You know, it's kind of a variation, which makes for interesting things when you're trying to get certain genes expressed in different organisms. When you're doing transgenics, you could actually make a bacteria produce a human gene, but if it's going to do so efficiently, you might need to change the sequence of the human gene to the codons that the bacteria likes to use, or else you might not be able to make very much of that. This is called codon optimization. We'll talk more about that when we talk about recombinant, but it just came to mind. So I've said that what really determines what the nucleotide to amino acid language is decided by the tRNA. That's not quite true either, because the tRNA is not what decided what got stuck on the end of it, right? The leucine being stuck on the 3' of a tRNA with this anticodon, the tRNA didn't moderate that, right? Something else, an enzyme, is what actually stuck and formed that covalent bond between the leucine and the tRNA. So these proteins are called tRNA synthetases. tRNA synthetases are proteins that grab tRNAs and grab amino acids and stick the appropriate two together. Okay? Well, I'll go through this, and then in the process I'll repeat it. So here's a aminoacyl tRNA synthetase. This guy is going to specifically grab valine. So the first thing that they do is grab amino acids, and they'll grab specific amino acids, right? So this protein specifically knows to grab valines. So it grabs a valine there in part of its active site. This requires ATP, so there's an ATP binding site. And then it's going to specifically grab a tRNA, and it recognizes the anticodon. Okay? So it picks the proper tRNA by reading its anticodon and sticking the appropriate valine on there. So you get that uncharged tRNA, the tRNA that's lost its amino acid. The synthetase binds it, sticks the two together using the energy and the phosphate from that ATP. And then I've got a charged tRNA. It's got the appropriate anticodon and the appropriate amino acid. So you have to have a lot of proteins and a lot of organization in order to translate a nucleotide language into an amino acid language, right? Because for every tRNA that I'm going to load up, I have to have a specific synthetase to do it, right? So we haven't even started talking about polymerizing them and putting them together properly. We're just talking about getting the raw materials to make a protein. There's a lot of stuff that's involved, right? I mean, that protein itself had to be made by the same mechanism that it's making the charged nucleotides, right? How do you make a tRNA synthetase? That's a protein that had to be organized, that had to be put together by tRNAs and amino acids put together by other things. Do you see the chicken and egg problem here? Yeah. How did you ever get the first nucleotide to make the first protein? You have to have a whole apparatus, like a whole host of people doing this in terms of molecular people, right? So this is a problem. This is in a way an irreducibly complex kind of thing that's going on in your cell, right? This gets into origin of life stuff, right? How could you ever originate this language without all the apparatus necessary to decide how to do this? All right, so there's my charged tRNA. It's stuck to the 3' end here. The last 3 nucleotides of all tRNAs are CCA. So here's CC, and then here's the A nucleotide. And it sticks this on, what is that? It's the base is always on the first carbon, so 1, 2, 3. Third carbon through is bonded to the carboxyl group of the amino acid. Okay, so here's your amino acid, your amine group, your carboxyl group. That gets stuck on the 3rd carbon of that A. So every tRNA has CAA, and it's stuck there on the 3'. Yeah.
So then do all those tRNAs and synthetases and all that stuff have to be replicated as well as the genome before mitosis can happen?
Yeah. Well, not before mitosis can happen. All you need is DNA polymerases, right, to copy. But yeah, when you divide, you better have tRNAs and tRNA synthetases and stuff already in the cytoplasm so that when the two cells divide, you've got all the apparatus to actually make your stuff, right? Yeah. I mean, that's part of the gap phases in the cell cycle, right? The G1 and the G2, it's making sure that the cell's got all the necessary stuff to actually act as an independent cell, right? Because you can't just take a plasma membrane and stick DNA in there and think you're going to have a living cell, right? That's not going to work. You have to have all these cellular components. So some people say things like, really misguided things like DNA is life. You know, that's just not true, right? Cells are alive and they use DNA to make their raw products. But you have to have a cell, right? Maybe there's an argument to be made for viruses being alive, but they are just alive because they're mooching off somebody else's cells, right? You have to have cells and proteins and all of this apparatus to get all this going, right? DNA itself is going to do nothing. RNA itself is going to do nothing. You have to have this intricate dance of DNA, RNA, and proteins already existing in a cell if you're going to have anything alive. So, which is a hard problem for origin of life. How can that evolve in very small little incremental steps? It seems like it's an irreducibly thing, the cell. Yeah.
That was actually my question, if you thought the cell itself was irreducibly complex.
Yeah, I do, yeah. I mean, some people disagree with me, but, and can imagine RNA worlds where you could have living, replicating RNAs on their own, but I find that hard to believe, so.
That's actually what I was taught.
It's part of why I'm a theist, is because I don't think you can generate living things just from natural laws. So, but all right, let's talk about, in the last couple of minutes, putting this actually together. So you've already got the language sorted out, right? The tRNA synthetases are what putting the amino acids and the tRNAs together. The ribosome just puts things in the appropriate order, okay? So it doesn't determine the language, it just reads and translates, okay? This is an amazing, amazing picture. This is the crystal structure of, I think it's a bacterial or a yeast ribosome complex. So the ribosome is a very intricate, very huge, multifaceted enzyme. It's got in blue here, this is RNAs, ribosomal RNAs, so specific RNAs that have been made off of the genome for the purpose of being in the ribosome here, okay? So all the blue is all RNA. In purple here, these dark purples, this is proteins. This is one side of the ribosome. It's in two parts, so here's the top part. Down here in orange, this is proteins. In yellow is RNAs. And then here in the middle, these are the tRNAs. There's 3 tRNAs in this. And can you see the template mRNA? It's not quite clear. I think it's one of these yellow nucleotides. This is an amazing thing. What this group did, and this is through, let me make sure I say his name right, Ramashrinan's group took this out of cells. They extracted a bunch of these out of cells, got them to crystallize together, shot X-rays at it, and from the diffraction pattern, they determined every single position of every single atom in all of these. So if you think about this picture, that's actually what the data is the atoms in the nucleotides of the RNA bonding together, and the atoms in the amino acids forming these structures, these looping structures. So this is a cartoon of actually an atomic level knowledge of where all the atoms in this whole complex are. Probably needless to say, he and his group won the Nobel Prize for crystallizing and determining the structure of a ribosome in action. I think it was awarded in 2009. But this is an amazing thing. We live at the cusp of knowing things that people have never known. I mean, we've known the amino acid code for years. I mean, Crick worked this out with Brenner in like the late 50s, early 60s, right? So we've known what the code is. We've known that ribosomes do this just in the context of if you take active ribosomes and stick them in cells and manipulate it, you can know what comes out. But to know actually the structure of this at a fundamental atomic level is just amazing. We have so much knowledge of what the cell looks like compared to people. And it's just beautiful too. I mean, it's really an amazing structure. So as an enzyme, we call it a riboprotein because it's a complex. It's an enzyme that's made up both of RNAs and of proteins, okay?
Biola University offers a variety of biblically centered degree programs, ranging from business to ministry to the arts and sciences. Visit biola.edu to find out how Biola could make a difference in your life.